this on? Hello, everyone. My name is Joseph Boland from the National Cancer Institute of the United States. So today I really want to take you through the past probably year and a half since we've had the PGM and shortly the Proton. So let's get started. Okay, so as I said, I'm from the National Cancer Institute. Within the National Cancer Institute, obviously, there are several laboratories. My laboratory is the Cancer Genomic Research Laboratory. Currently, we're comprised of 40 scientists and bioinformaticists. 30 of those are on the production genotyping side. So my section of the laboratory deals primarily with sequencing. We're 10 people right now. And as you can see on the slide, we have one of every instrument in the market today, high-seq, 454, and of course the PGMs. And that, oops, two things here. And this is actually one of the bays. And right now we have 6 PGMs, but we did start with 1. And so really I want to take you through what it was like when I had 1 and then the expansion into the next 6. So for today, as I mentioned, I just want to go through a historical review. So my section of the laboratory started 4 years ago. So at 4 years, we had nothing. And we've kind of built it slowly. And as you know, this sequencing market, it's rapidly advancing. It seems month to month at this point. I want to take you through the first impressions of the PGM from the early days through some of the presentations we made at a couple of the conferences in the US. Go through what we have right now, which is the PGM is actually one of our main confirmatory pipeline instruments for our exome or familial exome pipeline, and I'll explain that further. Take you through the first transformation, so the first expansion, and that happened at Christmas of 11 when we decided to go from 1 to 6 PGMs. The second year with the PGM, we started to really push it. We knew we can amplicon sequence with no problem. Now we wanted to say, okay, let's start exome sequencing. And with the rapid development of their chips at Life, there really was no point to kind of lagging behind. We could actually push this thing further. And I have data to show you that the exome sequence we did on the PGM really foreshadows what's going to be done on the Proton. And then really want to talk about the future directions and then what we're calling the second expansion. And that's with the Protons that are arriving probably within the next month or two at our laboratory. So let's go all the way back to 2008. In 2008, I arrived at this laboratory. I had 1 tech and an empty room, essentially, and tons of questions. Everybody wanted next-gen sequencing. No one knew how to do it at that point, at least in our laboratory, not in the industry. So we had tons of questions. We had investigators that wanted data the day before. We had several projects lined up. We had infrastructure items we had to deal with. And even today, when we bring in the PGM, it's not, it's almost plug and play, but there are some secondary items we need. We had to figure out what our staffing needs were. Was 1 person enough? The data storage, of course, over time that's now increased. And then, of course, the wet lab concerns with sample prep, sample quality, how do we quantitate the samples, how do you know you're putting 10 nanograms into an AmpliSeq study, and what confidence do you have? And then the bottom thing was to communicate between the bioinformaticist and the biologist. I'm a biologist, not a bioinformaticist, and that is a major problem in the US. I assume it's a major problem in Argentina. So that really needs to be explored, and you have to bring them in the laboratories, the computer people, as much as they hate it. You have to let them sit down next to you on the bench and really learn the technology of the science, and it really helps on the back end with the analysis. So in 2008, we had 1 sample flow. It was the good old days. We had 1 thing to do. Sample came in, sequenced it using the 454 process. And that was kind of nice, but we knew that wasn't enough. In terms of cost, the quality was very good, but cost per run was very high, and our throughput, could be better at that point. So we had to keep looking for improvements to the process. In 2009, really what we focused on was the front end, was the sample prep end. So while we kept just 454 sequencing at that point, we really wanted to look at what was the way to go about sequencing, regional sequencing or amplicon sequencing. Was it through traditional PCR, or was it through the Nimble-Gin, CCAP, so basically the hybridization? And then we were looking at different ways to fragment our sample to get it into the process, whether it was the nebulization process or the sonication. And obviously the sonication won out due to its throughput and its ease of processing. But this was 2009, so for the most part, it was still a single sample flow. But with this industry and everything sort of exploding, this is what we looked like in 2010. So we go from one process to several. And so we have all these processes ranging from short read amplicons all the way to whole genome. Now, I should tell you that for the most part, we outsource our whole genome. We're not staffed for that as of today. And all these processes feed our three instruments, Ion Torrent and then the two competitors. But I think what we're going to see in 2013 or late 2012, what we really want to do is pare this back down, not lose any of the quality, not lose any of our ability or flexibility, but really pare down these processes and pare down the instrument that we feed. So we don't have to train so many people on so many different platforms. And I think what we're going to see is really we're going to start feeding just the Ion Torrent systems, with the Proton being able to handle your high-level exome, potentially whole genome, transcriptome. You can really start to see, or I can start to see, phasing out some of the other instruments that we have. So I've had the pleasure to give a variation of this talk a few times for Life, and it's been a wonderful collaboration dealing with the folks at Life. And really the first talk that I gave was a first impression talk about what it was like the first day we got the machine. So in November of 2010, we had our first conversations with Life about this instrument. And obviously we were interested from the start, but we did have a few things that we had to go through, a couple thinking points or talking points that we go through for all new technology in my laboratory. So we want to focus on 4 areas. Excuse me. We want to make sure that if we replace an instrument, we're not going to lose any quality. It's a very simple thing. These aren't major issues or major topics, but these are things that we hold true. So it has to maintain or exceed quality. We have to see a path to reduce costs. So we can take higher costs in the beginning as long as we see a path forward to lower costs. It has to be easy to use. With retraining, while we're doing projects at the same time, you don't have that kind of staffing level that I can take 2 people off a certain project and let them focus for 40 hours a week doing training. So this is really what, one of the key drivers is it easy to use. And of course, can we reduce project time and, by which we can do more projects with the given staffing level, or maybe a slight increase. And so, sorry. So just to get to the point, with the PGM, we've seen is all 4 of these have been easily met. Now, for ease of use, I focus on that. As a 454 user, it was a very easy transition to Ion. That's not to say that if you've never done anything, you can't do it. Actually, it's quite the opposite. It's very easy to adopt. Just for us, though, it was very easy to read those protocols and move forward. The quality issue was, in the beginning, as Andy said, it was about a 2% error rate over 100 base pair read. We are now at 150 to 200 base pair reads and a sub 1% error rate. So, we've maintained the quality of 454 and started to exceed it as well. The project costs have been significantly reduced using Ion. Just to give you somewhat of an idea, I was looking at a project last night that if we had run the same project we ran 2 years ago with Ion, we would have saved about 75% of our costs. And then, of course, project time, it used to be in the past, all projects would be months. You'd have to think in terms of months, 6 months. Now we're thinking about days and weeks. So really, it has been a transformative platform in our laboratory and the number of samples that we run through our lab. So the first month, so just to give you an idea, with the PGM, we purchased it in November of 2010. It arrived January 2nd, 2011. On January 4th, we ran our first samples. I've never had a technology that was up and running within 2 days. I'm not just saying that because I'm here representing Life, but it's actually the truth. So for the first month, really what we want to do, we had one goal in mind with the PGM. We knew that, at that time, it was going to be an amplicon sequencing platform. And we had several hundred samples that we can envision running through. So like everything we do in our laboratory has to be multiplexed. We have to have chips that can handle several samples at a time. We can't do one at a time. It's just too slow for us. So we took the barcode sets that we had at 454, and using a platform from Fluidigm, went through and we wanted to challenge the system just to be able to differentiate the barcodes. So if we ran, as I did here in the first month, we ran a 1-plex. Could you tell me the barcode I ran? 8-plex, 24, and 48. And each time we would send the data to our bioinformatics folks, and then they would tell us which barcodes that they detected. And that's all we wanted to go after was the first month. Very simple, but very important at the same time. So this was what we wanted to do in the first month leading up to AGBT. Now, I'm going to reference AGBT a number of times. Some of you may or may not be aware. It's a conference in the US that for technology development folks, it's the Super Bowl of science. So every year we go, and it's always huge announcements. This year was Oxford Nanopore and the USB sequencer. I mean, it's always a big deal. So really what we wanted to do was be ready for that. So within the first month, we had 24 runs done. So here's a typical run of those 24. As Andy had stated before, our error rate was just around 2%. Let's see. Oops. Sorry. There we go. Total yield off the chip was about 28 megabases. At that point, Q20 was 4.65. It was a little raw at that point, which that's okay. In the research field, it's fine. And this is not the case nowadays, and I have some slides to show you this. So, and our mean length was right around 90 base pairs. So it was just about where we figured it would be. So for anybody that's bioinformatics inclined in the room, this is what we did to quality trim our reads to determine if the barcode really was there. We set the minimum read length to 10 bases because our barcodes were 10 bases. It was Gap-Smith-Waterman alignment. I couldn't explain to you what that is, but I trust somebody knows who that is. It's left anchored aligned, so we knew that on the 5' end of the read, that's where the barcode would be, so that's where it would look for. And then for all of the runs that we did, those 24 runs with the barcoding, we had over 98% of the reads aligned to the target, which at that point we thought was very, very good. So just so you can see, sorry about the small print here, on the left are a number of runs, 8 runs that we had with different plexes, 8-plex, 48, 16-plex, and the bottom one is actually blown up here, and this is a 16-plex where these are the number of reads, and you can see clearly that it's able to determine exactly what the 16 barcodes were, while really, it doesn't take, the bioinformatic guy knew right away which 16 we used and which 32 we did not. So we knew we were on the right track. So as a result, we submitted this abstract to AGBT. It was accepted. We gave a presentation. Everybody was happy. And really, it kind of launched us into, we knew at that point, the PGM was here to stay in our laboratory, and we could move it forward in our amplicon sequencing realm. So that was the first month. Obviously, it was nonstop. But since then, we haven't stopped. We've kept expanding. As I mentioned before, the PGM amplicon sequencing is the sole platform for our familial exome confirmation pipeline, and I'll talk about that a little later. Q20 bases are exceeding 150 bases. We've run older regional sequencing studies that we had done previously on the older platform compared to with the Ion with very, very good results. We've tested multiple software upgrades to the point now is that a biologist like myself can read the variants off of the PGM and actually trust those calls with great accuracy. And our head of bioinformatics is actually quite impressed with this. He still wants to double check me, which I would too, but he's very impressed with what it is. We've implemented an in-house 96-plex barcoding strategy. So that is in addition to the commercial 96. So we have the ability to do 192 samples per chip at a very high throughput level for us. We validated all 3 chips, 316, 318, and of course the 314. We have a bunch of one-touches and ES stations in our laboratory now working well. And then towards the end of 2011 into 2012, we ran our first exomes and transcriptomes on the PGM. Now, obviously, it's a little early for the transcriptome given the number of reads, but the quality coming out was very high. And then as of today, we're at 450 runs. And this is Dave Roberson, who we call Ion Dave. He's my tech, and we really actually love having him around. So the only question that we ask is, have we been successful implementing the PGM into our workflows? And I think the answer is a resounding yes. And I use this chart to kind of illustrate where we were. So this is 314 data. from the first month is in blue, and these were our typical yields, total bases, Q17 and Q20. So as you can see, it was rather low. And then roughly a year later, running the same samples across the 314 chips, you can see now where our Q20 data is approaching 30 megabases, which is fantastic because this represents how many samples I can run on a 314 chip, further lowering the cost of sequencing. Number of reads you can see are higher than they were, including the 50 base pair and the 100 base pair. And so back in January, this is really where we were, probably January, February, March. This was your 100 base pair chemistry. This was your typical mean read length and the number of reads. And by the end of the year, that's where we were. And so this 250 base pair reads really enabled us to start looking at exome and start really pushing it, as opposed to just doing small amplicons. And really, we saw a dramatic improvement in the consistency of the reagents. And of course, the user, because we've run this every day, and we got better and better. You really saw a dramatic improvement in data quality. So, no longer have we had, of course we have some problems with homopolymers, but not to this degree anymore. We very rarely see this, if ever. And it's very easy now. You can see this. And of course, when we started to see this, then you can obviously start making calls of samples and variants. You knew if you had clean reads, with pretty high confidence, if you saw a variant, it was probably there. So this is the second example, and this was a sample we ran early on that had a known SNP. And obviously, it was biased because we knew it was there. So we knew we could make the call, but obviously we couldn't put this in a paper and a presentation. No one would trust us. Everybody would laugh us out of the room with all this garbage going on. But now, obviously, very, very clean. You know exactly where the variant is. If I didn't know what this variant is, obviously I could make this call. So it was this steady improvement that really kind of gave us the impetus to move on and keep pushing the system in terms of number of samples through, as well as different experiments. So briefly, I could go on for hours for the 454 versus Ion. But this was one of our first 454 studies that we had done. And we took the same samples and ran it over on the PGM. And our concordances were around 95%. And going back and forth with the bioinformatics guys, you could make the argument that the errors are actually on the 454 side. But obviously we need to explore this a little bit more. But when we ran this, and this was probably mid-2011, this was more than good enough for us to justify moving away from runs that were 5 times more expensive to go onto the Ion. There we go. So these are snapshots. I always talked about barcodes in the beginning. So one of the nice things on the PGM, when you run it, you can upload your barcode sequences. It does the parsing for you. It gives you very nice graphs in terms of number of reads, number of Q20 bases for the barcodes that you have entered into the PGM, into the torrent browser. One thing I wanted to point out, obviously, these are the number of barcodes that we have. So this was a 16-plex, relatively balanced, which we were fine with. Number of reads exceeding 50,000 reads per sample. That's what it represents. And the one on the left that I have circled is X, and that's the one that either the barcode did not ligate on properly or there's errors. And so you can see what we want to see, obviously, is this be very low or minimal. So we're very happy when we see this because we know that our library prep process went very well. And so I just wanted to show you, give you an idea of the easiness to interpret data coming off the PGM. This is another one. This was a 47-plex. So I actually told Dave in my lab it was a 48 sample one, but I purposely left a sample out to see if the process was actually working well. So as you can see, this one did not report anything, which I was happy to see. Even at 47, you're starting to see really nice balancing, right around 70,000 reads per sample. And then this is a 58 one we just did, which is a little more choppy than we'd like to see. But when even your worst sample is around 35,000 reads, you're going to have more than enough data on that. And the other thing that I wanted to show, and I should have said before, is that this is AQ20 bases. So obviously, all the bases, high quality bases at that barcode position. And so you can see how it mirrors the barcode. So, and that's really obviously important for us. You know, our barcodes are built that it can withstand 2 errors and we'll still be able to separate out the samples. But obviously with high quality bases like that, that doesn't come into consideration. Usually it's, you know, 10 for 10 or 100% accurate for our barcodes. This was a chart that Life actually had published about their workflow improvements over 2011 from Q1 to Q4. And this being hands-on time from Q1 was about 3 hours. By the end of the year, you know, it was right around an hour. And total time was about 8.5 down to, you know, 3 hours of total time. And the only thing I wanted to show with this is that, you know, I think total time what we saw was slightly higher than what they reported, but certainly nothing that would give us pause. But the amount of hands-on time as we got better, you know, this is, you know, probably early days, you know, we're kind of learning where everything is, you know, was slightly higher than where they were reporting, but by the end of the year, we were exceeding most of their charts. So I think, you know, through their protocol improvements and through, you know, the text improvements, I really think, you know, you'll be able to exceed or at least match what Life's reporting for how long things take. So these are some of our current stats right now on the PGM. So we have perfect alignment reads ranging for our E. coli control, 256. Our human, 2 months ago, we hit 355 bases, and human RNA were about 210. The largest yields for Q20 for the 314 is 68 megabases, obviously not as good as Life R&D internal, but for us, this was you know, reason to jump for joy. This one was done a week ago, actually, almost 500 megabases on a 316, so that was a good one. And then the 318, we're right about to that gig. We're at 890 megs of AQ20, which raw was about a gig. So this one's improving, but we're happy with that. And the one on the bottom left here, I just want to show you is the median read lengths and how over the year, with everything, with all the new reagents, new chips, obviously us getting better and better, you can see the median read lengths just increasing. So, you know, first 5 months, 75 to 80 bases, you know, and then we just kept improving to the point where now we're at between 150 to 200 bases consistently on all runs that we do, you know, that have amplicons at that length. Obviously, if we run shorter amplicons, it will be shorter. So that was just the first year. So that was 2011. Obviously, it was really cool, and we had a lot of fun. And, you know, obviously, you know, we got a lot done in that year and really kind of showed the importance that this machine was having in our laboratory. So as a result, in AGBT, remember 2011, we put in 1 abstract about what we were doing. For AGBT 2012, we put in 4, and they were all accepted. And, you know, obviously we drank, we were happy. So the first one was exome sequencing on the PGM. We called it Exome in a Day, which got everybody excited, and I'll kind of go through what we did. The second was really transitioning genotyping to the PGM as opposed to traditional genotyping. The bioinformatic guys working with Life were working on new genetic variant software. And then using Kappa Biosystems, Dave in the picture there was working on a new way to quantify libraries before going on to chips, which would actually decrease the number of polyclonal reads that you were talking about. So really, after that year, we knew that we could move our confirmation or validation pipeline to the PGM. And really what we wanted to do was move what I call the exon hits or exome hits or variants from Sanger or Roche. And so what we did is we developed 2 workflows. And really those impact of those workflows was the savings of time, money, and an increase in output. So obviously 3 major things that we look for. And of course the 4th was ease of use that I didn't list here, but that's implied at this point. So there's 2 primary workflows for confirmation, like I mentioned. So the first was a sort of medium throughput pipeline. So I should step back and say the familial exome pipeline that we have, we don't do population-based exomes. We do familial-based. So these are disease burden families that have no known genetic cause. All the known genetic tests have come back negative. And so what happens is each one of these families, we exome 4 to 5 members per family. Those 4 or 5 members allow us to get a variant list, best case between 50 down to 5 variants, just to give you an example of the stretch. So obviously you could see the challenge of running that on other instruments. The cost and the time to design those primers, to run those would be very expensive. So using the PGM, we could do 2 methods and solve the problems. The first method would be using the PGM coupled with the Fluidigm access array. I'm not going to go into a lot of detail about the Fluidigm. I'm not sure how many people are familiar with Fluidigm down here. But really, this was kind of a marriage made in heaven from our perspective. They were both low-cost throughput, so this chip was right around $7 a sample. They were both extremely flexible, and I'll kind of go into why this is flexible. They both produce high quality, obviously, sample on this side and data on this side. They were both high throughput in terms of what our laboratory, obviously people's versions of high throughput can be different. And they were both relatively fast. You could do this process in 5 hours and of course this in under 2. So it was very fast. Especially one workday, you could have 48 samples by 48 variants complete. So as I alluded to, so this is what we were looking at in our laboratory at that time. So this was the new workflow we were talking about, using the access array, the one-touch, and the sequencing run. So this was for theoretically if we had 48 samples and 48 variants. So basically what happens is when you exome sequence a family of 4 or 5 people, the family itself is much larger. So we have to verify those variants. It could be a family of 50, it could be a family of 100. So depending on that family size, if we had a family, say, of 48 samples, and we had variants up to 48, you don't need to use all 48 slots. If you use the access array, what happens, and I'll take the next slide really goes into a little more detail for you. This is actually your library prep method all in one. So after amplifying your targets, you actually have amplicon targets ready for sequencing that have the ion adapters already on it. And then it goes into the one-touch and the sequencing. And you can do all of that in less than a day, and actually less than a business day. So if you started this at 8 o'clock, by 5 o'clock in the evening, you would have data. Compared to what we had at the time is if you ran through, if we did rain dance with 454, it would be 8 days. And then if we just did traditional PCR in 454, it would be 4 days. So as you can see, we were remember in the beginning I was talking about weeks and months. Now we're talking about weeks and days. And so this obviously is a single day. So obviously this is what we wanted to do. Just to give you a very brief explanation of access array. And, so this is the chip down here, and this is kind of blown up, and this is a individual chamber. So there's 2,300 chambers. So that's 48 samples by 48 variants. So each sample and each variant primer set go into an individual chamber. And there's a second universal primer. So the target primer has universal tags on it. It amplifies your target. I think, do I have that slide? Yeah, this is easier to do. Excuse the cartoon, but it's easy to explain this stuff when I draw it as a cartoon. Okay, so you load in your sample. You have your target-specific primers with your universal tag. What happens is during the first round of amplification, it creates your intermediate product with 2 universal tags. From there, a second set of primers come in with the universal tag plus the ion adapter and the mid built in. So you can pool from a 1-plex up to a 48-plex. You don't have to, it's not one or the other. You can pool as many samples as you want. After the second round of amplification, the product that you harvest out of that chip has your ion A and P1 adapters and mids on both sides. And at this point, you're ready to go into the one-touch for processing. So obviously, you can see that, for us, having the ability to do 48 samples at a time, obviously is a great time saver. The second method really is what we call the rapid confirmation. This is where, in the unlikely, but it does happen, some of these families actually have less than 10 variants, or in the best case, 1 variant. So typically what we do is usually outsource all our primer design. For anything less than 10, we design primers in-house. And so using the barcode strategy with the Ion library prep, we design the primer, we amplify it, we use the library prep, and then we run a 314 or a 316 depending on how many samples we have or how deep you want to go. I guess I haven't mentioned that. For the most part, we tend to oversaturate each sample just because we, with the fact that 314s are getting 50 megabases of information now. We're in the 1,000 to 2,000x per sample. And then we aim for 1 week turnaround for the client. They obviously don't know that we can do this within 4 days or faster. We can get the primers back from where we order our primers for. But if we could actually do that, then we could cut this down to a day. So what happens is we design the primer, send the design out, comes back in about 3 days, validate it, and then we go through our typical Ion library construction, one-touch sequencing run, and then the data review is rather involved at this point because it goes from me to the head of the laboratory. We kind of co-review it, make sure we're in agreement what it is, and then it goes to the PI for a final review. And all of that takes literally about a week. So in late summer 2011, we introduced this to all of our clients and all of our investigators, and this is what we've been using since. So instead of having 2 pipelines, depending on what platform and what kind of number of samples you want to do, now the PGM has really taken this over, and it's been really the workhorse for us. At this point, we've put about 3,000 samples through this pipeline, and it's been a wonderful experience because it's actually worked very well, either with the small number of amplicons or the large using the Fluidigm system. So, like everybody wants to see, this was the initial results. So after we put this in process, we had 10 targets identified from a disease burden family. 2 of the variants are shown. So these are 4 patients for 1 variant. So I'm sorry if I wrote that. So obviously you can see 1 has the mutant, the others are all clean, and you can see that this is very nice. You very little, if any, phantom eyes or insertions of wrong bases or missing a base. So we were extremely pleased to see this, and it made calling that much easier. This was the second 2 other family members with a different variant. At the same time, you can see nice bidirectional reads, nice balance there, and you can see the homozygote clear as day, right where that is. So of course, there's always work to be done with this pipeline. We're definitely pushing the system, so there will be about 5,000 more samples we're going to try to do by the end of the summer, probably into the fall. Of course, the early days was with 1 machine. Going in with 6 now, it really kind of redefines how we approach this, whether we're just going to process all 5,000 samples in library prep first and then just sequence them all in a single day. We just haven't really decided how we're going to handle that. We're still in discussions about that. We always want to look for new barcodes. Because of the new chips coming out, 192 is just not going to be enough on the P1 for the Proton or even the P2. So we are envisioning maybe a 500 or 1,000 plex run. And that's really where we want to be probably by mid-2013. We want to be up to that kind of plexity. Really, obviously, the read length is key. We want to be as far as we can. So we're assuming by the end of Q1 2013, we'll be at 400 base pairs. And then the bioinformatics folks always make me put this in, is that they're never happy with the software, even though we are. They never are. So they always look for ways to improve it, to break it, to fix it. That's just their job. So these are just some screenshots from the reports that we're getting off the pipeline. So this was a 316 run with different variants. So you can see the nice spikes of where the variants were. Our yield, obviously, the spec out in the market is 100 megabases. So for Q20 bases, we're doubling that right now and slightly exceeding it. And this seems to be now the norm. Now they may even have to rewrite their specs because it's well above 100. But this is always great to see, just so we know it's working for us. Your key peak value is 75, which we love. Anything above 60, we kind of take forward. It's just a good indicator of how good your run is going to be. This was another one the other day. So this was the record-setting one. I think, as Andy said on ion recognition, I think we're now 4th or 5th on the list with this run for a 316. So this was a really good run. We're not quite sure how we did it, but we'll take it. So as you can see, we almost had 3/4 of a gig of raw reads and half a gig of AQ20 bases. And you can see the nice distribution. Most of it is high quality 200 base pair reads. So that was the amplicon sequencing port. So really what now I wanted to kind of change topics a little bit. And currently in our laboratory, all of our exome work is done on the HiSeq. So obviously with the HiSeq, we can run many, many exomes. Problem being is that it's an 6 to 10 day run. So once your samples are on your flow cell, you have to wait over a week. I'm impatient. My investigators are patient. So we really wanted to see is there any way we can do this faster? For a number of reasons, and I'll highlight them as I get into this. So this was the talk that I gave down at AGBT for exome sequencing and exome in a day. So why exome on the PGM? One, because we could and we wanted to. Two was, we only have a single HiSeq. If it breaks, our exome pipeline is dead in the water. And it's used mostly for exomes. And really, there's no capacity for the R&D side really to get in to use it. Right now, this is kind of a little outdated, but right now we're at 48 to 56 exomes for every 6 to 8 days. So if we have a sample that drops out, so if you're watching that run, and if a sample does drop out, you have to wait 2 weeks. So really what we started to envision was, okay, maybe the HiSeq can be a workhorse for now, but the PGM really could come to the rescue if we have a sample drop out. We can get it right back in, and have it finished before the run finishes. So you can incorporate that data together and really because we're doing family-based, it is absolutely paramount that we have all 4 patients go together during analysis. If we're missing one, then your number of variants will increase because you just don't have that, those 4 family members to really look at heritability. So this is what we want to do in the PGM. Give us some flexibility. So the real-world applications, anytime a manuscript is written, if a reviewer wants more data, that's not going to happen using a HiSeq. It's going to happen using a PGM. We can actually give you some exomes, safely within a week, obviously shorter than that. If you have an impending site visit, so at the NCI, we have audits every 4 years, and we have to make ourselves look good. We have to have data. We have to have projects completed. So if we have projects that have holes in it because samples failed, we have to have ability to get them back in. This is the one that we're going to highlight in a few minutes is what happens if you have one family member fail. Obviously, your analysis has to wait for that. And this is where I really wanted to push it to see, okay, could we actually do this? Could we take a data set from Illumina, wrap in a PGM sample? And then, of course, primary machine downtime. We had to have a way, if it went down, just to kind of limp along until it was fixed. And this was all done with the PGM. So obviously, what Andy's saying with the 2 exomes per day on the Proton, this was just whether we could do it. And now moving forward on the Proton, obviously, we know we can do it. So here's the workflow that we came up with. Okay. So library prep is equal in both pipelines, about a half a day. Sample captures 3 days, same thing in the PGM. Their CBOT's half a day. Template prep is about a day. And so all told, this was 8 to 10 days for a set of exomes. In the rapid pipeline, it'd be 1 to 2 days per exome, depending on the number of machines you have, and I could talk about that. And so obviously 4 or 5 hours runtime, 4 to 6 hour process time, about a half a day for the bioinformatic folks to go through. So it's faster on this side than obviously, so it's 8 to 10 days just to generate the exome, but then it's another 2 days to crunch all that data, to really create all the BAMs, all the alignment, and figure out what you actually have. So for the library prep for this, we use standard HapMap samples, and these are the numbers in case anybody's interested in getting them for their laboratory. We followed the protocol that Life has at that point to a T. We just didn't want to disrupt anything at that point, so we just wanted to use something that worked. We used their target-seq exome capture. It is not the one that Andy talked about. This was an earlier version. So this was, I think, about a 37 meg. I don't know if it's listed on there or not, but it was quite a bit smaller than the current one at 50 megs. And we used half a microgram of sample, and then the hybridization was about 65 hours. And then obviously this is the adjuvant tracing of the exome peak after capture. So we knew we had a nice nice good peak. These are the reference peaks with no garbage left or right. So we knew the samples were of high quality. So these are the samples that we used. It was actually a pedigree 1463. So dad, mom, and son, just for ease of presenting. It was a collaborative data generation. So at that point, we ran, just for this part, we ran samples here at Ion East and West in the United States. So all three labs kind of worked together to give us some data sets. The question was, how much data is enough data for exome? It's always been a big question because obviously you know what you can get on the other instruments, but is that too much? Are you just wasting space? And so for mom, what we did is we generated data sets of 3.5, 5, 7, and 10 gigabytes, just so we can see, was there a point where it was just too much data, got nothing in return at that point. And then dad and son, we ran at 5 because we knew 5 was the answer, but that was we ran it after we looked at mom. So just to give you an idea, what we shoot for when we sequence exomes is we shoot for 80% of the exome covered at least 20x depth. So as you can see, based on the lines, all three, with the exclusion of 3.5, exceeded that boundary between 80 and 20. And the 3.5 was slightly underneath. Still usable, but just slightly underneath, right around 18%, or 18x at 75%. So we knew this was probably a little underloaded, so this is why we went with the 5, just because at this point, this is what I was told by the bioinformatics guy. So we went with 5 for dad and son. So this is some data that I might stumble through, so just give me a minute here. So this is what we did for mom, is we had the 3 PGM runs of 3.5, 5, and 7, compared to the same sample that was run in a high-seq, compared to Complete Genomics whole genome data for that sample. So for this, we used Complete Genomic as the gold standard, as the whole genome, relative to the 2 next-gen platforms. So what we wanted to see, is a nice, do you see any kind of correlation? Does one platform or another better than the other? What we saw, surprisingly, is we wanted to be in the neighborhood of Illumina. Turns out we were in a better neighborhood. So, they had a 6.5% novel rate, which is way too high. Most of those are probably false. Versus the novel percentage, what you theoretically want is somewhere around 2. So we were very happy to see that we were at 1.7 to 2.1. So, we were more surprised at ourselves that we could generate this as opposed to, wow, looks really good. And then complete, of course, was 3.4, but that's the whole genome. So we didn't, we're not going to say ourselves are better at this point than complete, but we're working on it. The ratios, the transition to transversion ratios, for known mutations, you want to be around 3. And so that was all fine. For your novel, you want to be about 1.5, somewhere around there for a ratio. That's why the high-seq was 0.3. So that tells us that a lot of these novels are truly false. So obviously for the total, you want to be in the 3 category. So we were happy with this. Just a little background information. As I said, complete was the gold standard. The variant calls are based on the coding sequences of the UCSC known genes. The known variants are based on DB SNP 135 plus 1,000 genomes. And just to be fair, Illumina was generated on Nimblegen version 2 exome capture, which was one behind what we were currently using. And then on high-seq, and they use NovoLine and GATK, or GATK. So the genotype discording rates, just to visually show you, obviously at 3.5, it was a little higher than we'd like to see. Not terrible, but certainly we knew we can go better just by adding 1.5 gigs more data. So for our discordance rates, we're around 1.5 for novel. For known variants, sorry. And then for all variants, it was about 2%. Relative to the dad and son at 5, you can see where the dad and son using just 5 gigs really fell nicely in line with mom's rates at 5 gigs. So just so anyone who's interested, the Mendelian inheritance rate of the trio was right around 3.88%, which is slightly higher than we'd like to see, but certainly nothing alarming. Okay. So, we use the trio as a reference. could we exome sequence? Had nothing to do with speed. We just want to make sure that the quality was there and that we could make calls. So at this point, we wanted to challenge ourselves and say, OK, let's do a real-life situation. So we have a melanoma-prone family. Diagnosis is at 38 compared to 60 years old in the US population. It does suggest an autosomal dominant inheritance with incomplete penetrance. And so what we did is we had 5 members with cutaneous melanoma. We exome sequenced the 5. So what we did was, OK, what happens if dad fails during the exome? So let's see if we can recover it and produce data that works. At the same time, we had kind of a bonus. We had a matching tumor slide of dad. And so we had one slide to use. It was 10 years old. And we were able to use the cancer panel to kind of profile the tumor at the same time we were doing exome. And I'll talk about this in a little bit. So we targeted exome in a day. We targeted 5 gigabases. So we wanted to figure out if we started at 8 a.m., by 8 a.m. the next day, could we have data to the bioinformatic folks already churning and burning? So obviously, this part takes longer than a day. So it's obviously with an asterisk, but this takes about 4 days to get it prepped, do the capture, and then 1 day. So really, after this is really when the clock started. Obviously, I know it's biased, but this is what we wanted to do. So we wanted to go 5 gigs. Using 318 chips, we set up 5 chips. So obviously, 1 chip per machine. Now, I will tell you that you can do 2 chips per day per machine. So if you have 1 machine, it takes a couple days. So just because we have 6, we can do things like this. It does not preclude somebody with a single PGM to do this. It just takes a little more time. So it's 4 hours runtime per machine, per chip. The process time was about 4 to 6 hours, closer to 6. The BAM files and the variant calls were about half a day. The AmpliSeq panel took an hour and a half to run on the machine. And the variant calls took less than an hour. I think we timed out to about 45 minutes at that point. So this is what we wound up generating. We got 4.8 gigs of sequence. So it was right around 8 to 900 megs on per machine. We had 19,000 SNPs, very much in line with our HapMap trio, which was very nice to see. Our novel variants came in at 1.9%. Our ratios were very, very much in line with the 5 gigs that we saw in our reference, right around 3. And our novel rate was 1.5. So we were fairly confident that the novel mutations coming in were in fact true. And then comparing the variant SNPs with CGI data for this sample, we had about a 2.2 discordance rate. So at the same time, as I mentioned, we ran the cancer panel for the tumor. We wound up generating 200,000 reads. We had 7 million map bases. Percent basis on target were about 58%. It's a little bit lower, but this is FFP. It was our first time isolating it. So obviously, it wasn't nearly as good as it could have been. However, when you're looking at the target region, because the bases in the target region were only 13,000 bases for the cancer panel. Average coverage depth was about 300x. Uniformity coverage was about 71%. So what that really tells you is that at 1x, greater than 99% of the bases have a call. At 20x, it's about 84%. And then you really start to see a dive at 100x for about 60%. So we wound up calling 109 hets, 10 homozygous SNPs, and then 1 indel. And just so you're aware, we had 1 slide. We used Ambion's Recoverall for the extraction. And then we ran the 1.0 at the same time as the exome. So we utilized all 6 machines exact same time. So the results of those 116 variants, we thought the caller at that point was a little too sensitive. Most of those actually came out in post-processing. So we had 26 variants left that we felt were true variants. We confirmed germline for 16 of them, whether it was in the exome or it was in complete genomics data. On the positive side, we found 10 potential somatic variants that we further validated as present in that tumor using another slide of that same tumor. So I guess the last section I really want to talk about is the cancer panel and the custom AmpliSeq panel. Obviously, we use the cancer panel for our exome tumor sample. And we've wound up using this panel a number of times for, while we're not a clinical lab, we're a preclinical laboratory, research laboratory, but we have a lot of sister labs that are clinical. They have a number of tumor samples that we'd love to get our hands on just so we can either validate what they have already called using traditional methods for mutation analysis, or give them an added option that we can profile samples for you. And then you can develop something clinically based, but for fewer variants. And I have some data for that. So we've used the hotspot panel several times to validate some somatic mutations, being able to detect down to 5%. We recently just ran the comprehensive panel for 4 tumor normal samples for renal clear cell carcinoma. And that data is actually under analysis right now. And then using the AmpliSeq design engine that Andy talked about that hopefully will be available to you soon, we've put about 1,000 primer pairs through that. And I'll talk you through that, how we've used it and what we like about it, what we don't like about it. So for the hotspot panel, we ran 7 tumors. And these were the results that came out. We were very happy with the allelic balance. So if it was a het or homozygous, it really was either 50 or 100% or very close to it. The last tumor actually had a tumor mutation down to 5%. We were very happy we detected that with great certainty at that point. It was 4,000x coverage at that site. So we were very comfortable calling that indel essentially. So for the comprehensive, before I get into this, the comprehensive, while it's 4 tubes combined into one on a 318 chip, we're not at the spec that Andy was talking about, and I think that's just getting used to the instrument or to the actual panel itself. We typically use 30 nanograms as opposed to 10 per tube. It's just a comfort thing, and it comes in with quantitation. You know, when we're at that edge of 10 nanograms, I get a little uncomfortable actually able to quantitate that accurately, so I always go up a little bit more just to kind of buffer that out. But it ran well. The 4 AmpliSeqs, you know, that takes 2 hours. You pull it together and you go into one chip. So it's actually quite streamlined, and I'm looking forward to actually getting into the data when I get back. So this is just a screenshot of the engine itself. Andy had a nicer one, but this is the actual dashboard. So when you log in, all of your experiments that you've put in to the design engine come up on what's called the dashboard. Now, you can order them after you see this. You can get them, not like them, resubmit. It is that easy. So what happens is it comes in, you just call it a name, whatever you want, load up the number, either genes, coordinates, you know, whole regions, if you will. And then what will happen is after it's designed, it typically comes back within a day, especially if you're doing just genes. If you load up 5 or 6 genes, one of these designs that I put in, it was 5 genes, and I had a response back in 30 minutes because I think they were already designed, so it was very easy for them to pull it apart. What happens is it comes in number of targets that you put in, and so if it's genes, it kind of matches the number of exons, unless the exons are very large, and it'll be number of targets per that. You can choose 200 base pair or 150 base pair. So if you have FFP samples that you think are highly degraded, you can actually go lower. And then it comes in, as Andy mentioned, with the number of primer pools. So 1 or 2 or 3 or 4. I've only had 2s. I think I had 1 there, but no, only 2s. And then it kind of gives you the % covered. So this is obviously key for us. For the most part, everything's been 98% or better. So we typically don't worry about the last 1.2%. And then it tells you the different pipeline versions of software. So this will change automatically when they update it. And then it tells you when you modified that project. So you can track it internally as to, did I submit this, did I not submit this? And then on the, sorry, I can't read it myself. So on right here on the order status, it actually says results only or ordered. So this is what I would imagine you'll see in the future. So what happens is it comes back, you get a bed file, you can put it in IGV, you can really see where the primers lie. If you like it, you just click order. I mean, it really is that simple. And then, like I mentioned, we've ordered about 1,000 primers. We've tested about 500 of them, and they are working quite well. Our fear was that the cancer panel, honestly, was going to be over-optimized for Life's products, that it would be this good, and then the custom would be that good. But no, actually, the custom has actually come in just as good as the cancer panel. That's why we've been pretty impressed with it. And based on what I said before about the 2 pipelines, whether it's the medium throughput with with Fluidigm or the low one with hand design, you really see using this engine now that I can actually get away from 2 and just go to 1 and then design it based on the number. So it's actually, for us, would be quite helpful. The best thing about this is that it comes in the tube pre-mixed, which is nice to get going right away, but it comes with the plates as well. So really, if you have, if you're lucky enough to have it by gene order and someone comes in with a variant and you just have that gene, you can just pull it out and run it singly very fast. So that's been quite beneficial for us. So this was a recent result we had with our first custom AmpliSeq, going after a number of targets. Obviously, we put a couple of control samples in here that we knew had nothing. But I think what we were most impressed with is that after running this really mirrors what we were seeing with the cancer panel. And I'm sorry if you can't read it. That's my fault for doing that. But the allelic balance is very, very nice for us. So this really gives us the confidence that what we see is, even if it's a major indel or it's just a simple SNP, based on the coverage we're seeing, as I said before, I think the number of samples per chip is we're too low. So we're really blowing out the samples to the point where we have some samples that are 32,000x. I think it's a little too deep. We can dial that back. But really, when it comes to these family validations, because the cost of sequencing is so low, it doesn't matter. You can run one chip with 5 samples. I don't care if I have 50,000x because it's so low. Cost is, at that point, is pennies, which for us is what we want to be. So that's been a really good thing for us. And even some of these samples that maybe the sample doesn't amplify as well, you're still at 45x coverage, which for germline variants, very easy to make a call off of that. So I guess the question is, where are we going? So probably about mid-2011, before the Proton was announced, we really envisioned us just keep buying and buying these things and having 40 or 50 of them. Thankfully, they came out with the Proton so that we don't have to have major space to have all these pictured. So what we are going to have going into 2013 is we'll have 6 PGMs. And these PGMs will be our workhorses for our exome follow-up, for any sort of regional sequencing that we have, any kind of small RNA. So even though we are a germline shop and a DNA shop, our investigators more and more are asking for RNA work. So instead of outsourcing it, we're going to now bring it in-house. Because these machines are flexible enough, I can run 3 DNA studies and 3 RNA studies at the same time. It's a wonderful thing. And then, of course, with the protons coming in, for us, sky's the limit. We're going to take our rapid exome pipeline. We're going to move that right onto the Proton. And that'll be 2 exomes per day per machine. So we will have the capacity to do 8 exomes, at least at the outset, in our laboratory per day. Whole genome sequencing, currently we outsource that. So obviously, looking at 2013, we want to bring that internally. Really no reason why we can't. Same thing with whole transcriptome. And as I mentioned before, really we want to go after high throughput amplicon sequencing. So we have these very, very large families that are 500, 600 members. We would love to do it in one shot. Don't have to process it once and you're done. And then, of course, single-cell genomics, which, this was really just on the drawing board right now for us. So I don't have a lot to say about it, but we know it's there and we know it's coming. So we're going to plan for that. So really the final thoughts, the PGM platform really has transformed our laboratory. At the point we bought the PGM, our costs were very, very high. We didn't know if we'd be able to even stay in the game based on our funding. But that really has helped us. As I mentioned, with the PGM and soon the Proton, our flexibility has doubled and even tripled of what we can offer while maintaining high quality, a low cost, and a high throughput. So the next steps, obviously I mentioned, we're going to push the amplicon pipeline with about 5,000 samples. The next few months will be establishing the Proton, getting that in, challenging the exomes that we already have done on the HiSeq, on the Proton, making sure that we're in the same ballpark, if not better. And then, of course, start to approach the $1,000 genome by Q1 of 2013. That's a goal of everybody's, and I think we're starting to see that's becoming a real possibility. So obviously, I don't do the majority of the work. None of this is possible without this laboratory here. This is the entire lab. Life was gracious enough to give us all Ion shirts when we bought the machines. So Dave up here has been irreplaceable. Same with Mike Cullen. They really have helped us go from a lab of 1 now to a lab of 6, and we're really driving the sequencing. So Thank you for your time, and if you have any questions.